Let's talk about some genetics problems because the genetics chapter is presented in a very problematic way. You have to figure out what kinds of offspring are going to be produced from specific parents. So let's take a look at this first problem. In pea plants, red flowers dominates white flowers, and we're going to use big R for red and little r for white. Tall, big T, dominates short, little t. Axial dominates terminal. Axial's big A, terminal's little a. Three different traits. The first problem here, cross a white flowered pea plant with a heterozygous red flowered pea plant. To get the parent genotypes, the Punnett square, a GR, and a PR. So we got a white flower with a red flowered. Well, here's the solution down here. The white flowered plant is little r, and the heterozygous red flowered plant is big R, little r. That's step one on the rules for Punnett squares. Step two is you need to figure out how big your Punnett square is going to be. Well, since this plant only has little r's, it's only going to produce a little r gamete. But this plant has a big R and a little r, so we can make either big R gametes or little r gametes. So we're going to have a 1 by 2 Punnett square, and here it is. We could have had one column and two rows. It wouldn't have mattered. But we decided to do a 1 row, 2 columns one here. And the white flowered parent, right, there's its gamete right there. And the red flowered plant, there's its gametes there. When you bring down the column headings and the row headers into the grids, the first offspring is big R, little r, and that's a red flowered offspring, and the second offspring is little r, little r. The genotypic ratio for this Punnett square is 1 of the heterozygous red to 1 of the white, little r, little r. So this is the GR. The PR is one red flowered plant to 1 white flowered plant. Okay, so that's the first problem. Now we go back to the top. It says cross a heterozygous red flowered heterozygous tall with a white flowered short. Okay, now we got two traits now. It wants the parental genotypes, the Punnett square, the GR, and the PR. So now this is a little bit bigger problem. The first plant was heterozygous for both traits, so there it is. Big R, little r, big T, little t. The second plant was homozygous recessive for both traits. So it's little r, little t, little t. Notice that this problem yet does not include the axial terminal. Okay, don't have to worry about that. How big is the Punnett square going to be? Well, there's 4 combinations of r's and t's that you can make from that parent. Either big R could go with big T, big R could go with little r with big T, little r with little t. Notice there are no sperm combinations where it's big T with little t or big R with little r, because according to Mendel's rules, each sperm has to have one of each factor type. So the factors separate, and a sperm gets one of every factor variety. So it has to have a flower color, it has to have a height gene. It cannot have 2 flower color genes, it cannot have 2 height genes. The other plant, the little r, little t, it can only produce one combination of r's and t's. That's little r with little t. So when you fill in the Punnett square, you get 4 possible offspring. And it's one of each. The GR is one of these, to one of these, to one of these, to one of these. And the PR, and that's what they look like, 1 red flowered tall, 1 red flowered short, 1 white flowered tall, 1 white flowered short. And that's the first, or letter B is completed. Now letter C says a heterozygous tall, heterozygous axial is crossed with a short terminal flowered plant. Right? Well, that's very similar to the problem we just did, except that here we got A's instead of the R's. Okay? So the second plant's recessive for both traits. The first plant is heterozygous for both. Well, we're going to end up with a 4 by 1 Punnett square, or a 1 by 4. Okay? And it's the same basic pattern. Here's all of the gametes from the heterozygous parent on top. Here's the gametes from the homozygous recessive parent on the side. And here's all the offspring from the cross. Notice we have again one tall axial, 1 tall terminal, 1 short axial, 1 short terminal. That's here. The GR is literally listed right here in the Punnett square. It's right there, one of each. You have to have the numbers with them. Now, the next problem, let's see, going back up. Bug problems. In bugs, brown body dominates white body. Long hair dominates short hair, and the trait is sex-linked. That means the L's are on the X chromosome. Size exhibits incomplete dominance. Huge is incompletely dominant over tiny. The heterozygote is medium. Aha. So we have a huge heterozygous brown bug is crossed with a medium white. Okay, huge heterozygous brown, medium white. So let's go down to the solution. Huge heterozygous brown, medium white. Okay, why huge? Huge here is big H, because huge is incomplete dominance. Heterozygous brown, right there. Medium is big H, little h, and white is little b, little b. So how big is the Punnett square going to be? Well, there's two different combinations of H's and B's for the first parent and two different combinations of H's and B's for the second parent. So the Punnett square is going to be a 2 by 2. And our GR is going to be one of each of these. One big H, big B, little b. One big H, little h, big B, little b. One big H, little b, little b. One big H, little h, little b, little b. And the PR that you have to interpret from those is one huge brown, one medium brown, one huge white, one medium white. Okay, so that's the first problem. Now the second problem is a little more complicated, and it's a bigger Punnett square. In letter B, it's got a medium-bodied, heterozygous, long-haired female. Now that means she can make 4 different gametes. You'll see in a minute. It's crossed with a tiny-bodied, short-haired male. Tiny-bodied, short-haired male. So let's scroll down. So here's the tiny-bodied, short-haired male. Notice that it's X little ly. There's no letter on the Y. Okay. Here's the medium, heterozygous, long-haired female. She's got X big L, X little l. Now this Punnett square is a 4 by 2. This is a big one. And that's because the female can produce 4 different combinations of H's and X's. The male can produce only 2 combinations of H's and X's. So that's why it comes out to a 4 by 2. Please remember, the Y chromosome gets no L. There is no gene for hair length on there. If you put that on there on your problem on the test, out of a 10-point problem, the maximum score you can hope for is 5 points. Maximum. And that's because you are disregarding one of the most significant contributions of 20th century genetics, and that is that males don't possess a fully diploid set of genes because of sex linkage, which has major implications on health, male health, for many males that are born with incorrect X chromosomes. So here's the GR, it's big, it's one of each of those. And here's the PR, it's big, it's one of each of those. I'll let you look at those more specifically on your own, but I want to keep covering the problems here. Letter C is a huge brown body bug who had a parent with a white body, is crossed with a tiny white bodied bug. So if we come down here, a huge brown bodied bug who had a parent with a white body. Now, that's basically stating that they're heterozygous for body color. They have to be. And that's because one of their parents, the parents of this bug, had white body. And if that's the case, then that little b came down here for sure. The other parent was tiny and white-bodied. And there it is. Now, this Punnett square is a lot smaller. This organism can only make 2 gametes. This one can only make 1. So it comes out to a 2 by 1 problem. The GR is one of each, a medium brown to a medium white, and the PR is 1 medium brown, 1 medium white. Okay, so that completes those Punnett square problems. Now we go to some pedigree problems. A normal man and a normal woman marry and have a son with CF. Draw a pedigree. Well, if the man and woman are normal, but the child has CF, that means that mom and our dad and mom are carriers. So they have to be half shaded. And the son has to be fully shaded because he's got CF, and that's a recessive condition. You always shade in the recessive genes. So this is like big A, little a, big A, little a, little a, little a. Okay. Next problem. A man with type B marries a woman with type B, and they have a daughter with O. The man's father had O, and the mother had type AB. Draw a pedigree. Okay. So here's the man, here's his wife, and here's the child. And then we tell you that this man had a dad who was O and a mom who was AB. So mom doesn't get any shading because her two genes are dominant. She's capital I superscript A, capital I superscript B. The husband, or the I could say the grandfather of this child, is little I because he's type O. For sure, then, this husband has the little I gene, so he's half shaded, but he's type B, and that means he's I, capital B, little I. Okay. His wife has to be, she's type B also, but she has to be carrying the recessive O gene because they have a child with O. So she's capital I superscript B, little I also, just like her husband. And then here's their daughter. She's little I. You can see that a recessive gene came from both parents to produce that blood type. All right, so that brings us now to the next problem. Eye color, which really is more complicated, but we're going to boil it down just for genetics problems. Let's just say blue is recessive to brown. A heterozygous brown-eyed person marries a blue-eyed person. Develop parental genotypes of Punnett square, a GR, and a PR. So what kind of kids can they have? Let's see. Here we go. There's the let's see. In the eye color problems, the one parent's big B, little b, the other parent's little b, because that other parent has blue eyes. They have a possibility of two different children. Their genotypic ratio is either their kids are heterozygous brown or blue. One brown to one blue is what you'd predict for the offspring. Number 5, color blindness problems. A normal vision woman whose father was colorblind marries a colorblind man. Okay, what's going to happen to their kids? Normal vision woman. Okay, here we go. Let's see, right here. A normal vision woman, here she is, but she had a colorblind dad. That means she's carrying the colorblind gene for sure, because dad gave her one of his only X chromosome, which was the colorblind X chromosome, because he was colorblind. Marries a colorblind guy. Okay. Now this mom can make two different kinds of eggs. Dad can make two different kinds of sperm, so you get a 2x2 Punnett square. The genotypic ratios are one of each of these. That's one normal girl, one colorblind girl, one normal boy, one colorblind boy. And there's the phenotypic ratio right there. Okay. Next color blindness problem. A colorblind man marries a homozygous normal vision woman. What's going to happen in their family? So here's the colorblind man. Here's his normal vision bride. She can only make one kind of egg. He can make two different kinds of sperm. So there's his sperm. Here's her egg. The ratios in the offspring are 1-to-1. Daughters are going to have normal vision. Sons are going to have normal vision. One normal girl, one normal boy. One X big N, X little n, one X big N, Y. Okay. Next problem. We got some blood type problems. Number 6. A type O woman marries a B man. They have an O son. Okay. Type O woman marries a B man. They have an O son. Automatically, that means that the man is carrying the O gene, because that way the son would be receiving an O gene from each parent. Okay. This is the pedigree. Dad's half shaded. Mom and son are fully shaded. And in the second problem, we have parents are heterozygous AB and homozygous recessive. So let's look back up at the problem. A type AB man marries an O woman. What are their possible children? Okay, so let's go back down to this. Here's the AB man. Here's the O woman. They're either, now we use diamonds to represent boys or girls. They are going to have either A children or B children, depending on which of these two genes gets passed down. This parent's always passing down little I. There's no chance for O children because the dad does not have a recessive gene to hand down. Now, number 7. Gene linkage. Two genes P and Q are 24 map units apart. Another gene R is part of another linkage group. That means it's on a different chromosome. If an organism heterozygous for P, Q, and R is mated to a triple homozygous recessive, what are the chances of an offspring homozygous for P and heterozygous for Q and homozygous for R? What are the chances of an organism that is recessive for all three traits? Now, this one gets kind of complicated, so let's go back down. I think I have it all worked out for you. All right. The linkage groups of this parent are the dominant P and Q are linked together. In order to and they are 24 map units apart. So that means that 24% of the time crossing over will occur, and big P can end up with little q in an offspring, or um, big Q can end up with little p. Okay, so the chance of little p ending up with big Q uh, and getting the little r is the chance right here, the chance of uh, little p ending up with big Q. What's the chance of that? It's half the crossover frequency. The crossover frequency is 24%. That because the genes are 24 map units apart. That means that 24% of the time it's going to be big P with little q or little p with big Q. Well, it's 12% for each of those. So it's 12% times what's the chance of being little r? 100%. Times the chance of being, let's see, oh, I'm sorry, no, the chance of being little r is 50%, because if I'm not mistaken from the previous problem, where is it? Going back up to the problem, the first parent is heterozygous for all the traits. There we go. So what's the chance of an offspring homozygous for P, heterozygous for Q, and homozygous for R? What's the chance of an offspring is recessive for all three traits? So going back down to the problem down here, it's worked out correctly. The chance of getting little p with big Q, Q is 12%. The chance of getting little p with little q is 100%, because that's all the other parent has to pass down. Crossover or not, little p is going with little q from the other parent. There's a 50% chance of being little r. The other 50% of the offspring are big R, little r. The answer to that comes out to 0.06. Now, the chance of being with little p, little q from the heterozygous parent, that's 38%, which is half of 76%, which is 100% minus 24%. So again, if the chance of crossing over is 24%, the chance of not crossing over is 76%. And that means that 76% of the time you're not going to get any crossing over. And in that chance, you're going to either have little p with little q or big P with big Q. And in this case, it's little p with little q. Well, it's 50-50 for that. So half of the 76, or 38% chance of this, 100% chance of that, because the other parent is recessive for everything. And the chance being little r is 0.5. So we'll talk more about this in class, but we only touch on gene linkage very lightly. Number 8, it says, suppose students flipped 4 coins to simulate the inheritance of traits, and they decided to simulate this cross, which is the dihybrid cross. They marked their coins A on the head side, and big A on the head side, and little a on the tail side, and the second coin was marked big. Oh, okay. So they marked 2 coins, big A, little a, heads, tails, and 2 coins, big B, little b, for the second trait. So they're flipping 4 total coins. Now, they flipped them, and their coin toss results are here. They had 435 cases of either of these 4 possibilities. They either had all capital letters, or almost all capital letters except for a little a, or almost all capital letters except for a little b, or a capital A and a capital B plus a little a and a little b. Okay. Now, they had 164 times they had 2 capital A's and 2 little b's, or capital A, little b, little b. And then 153 times they flipped, they had, in all cases, little a, but with big B, or with big B, little b. That's 153 of those. And lastly, they had 48 of all little letters. The question is, were the coins flipped randomly? So you are asked to do a chi-square analysis on this problem. So down below, I've got it worked out for you. First off, you're supposed to state the null hypothesis. And then your null hypothesis basically is that there will be no statistically significant difference between the observed coin flip data and the expected coin flip data. Now, you have to get your expected amounts, you have to add up all your tosses and then figure out how many of these you were supposed to get, how many of these you were supposed to get, how many of these, and how many of these. Well, as it turns out, 435 plus 164 plus 153 plus 48 comes out to 800. And it turns out you should get 9/16 of that should be the dominant for A and dominant for B offspring. And so 9/16 of 800 comes out to 450. 3/16 of 800, which is what you expect for this, comes out to 150. That's 3/16 of 800. Another 3/16 of 800 comes out to another 150 here. And you're only expecting 1/16 of the offspring to be recessive for everything, and 1/16 of 800 is 50. Now, to do the chi-square analysis, you need to figure out the observed and the expected differences. So the difference here is 15, the difference here is 14, the difference here is 3, and the difference here is 2. Then what you do is you square those differences. So we got 225, 196, 9, and 4. Then you take this equation, you divide this by what you expected. So 225 divided by your expected, which is 450, comes out to 0.5. 196 divided by 150 comes out to 1.3. 9 divided by 150 comes out to 0.06. And 4 divided by 50 comes out to 0.08. When you sum those up, it comes up to 1.94. Now, you were tossing 4 coins. By definition in chi-square, the degrees of freedom are always one less than the number of possibilities. Well, there are 4 possible coins, so the degrees of freedom is 3. So you look in the 3 column here, and you compare your number to this column of numbers. These are like steps on a ladder. The number 1.94 falls between 0.35 and 2.37. What that means is that you are between 50% certain and 95% certain that those coins were flipped according to chance events. And our conclusion is that the null hypothesis was supported. There was no statistically significant difference between observed and expected value. So there are 3 degrees of freedom because there were 4 possible coins flipped. Using the chi-square table, the chi-square value of the test must be less than 7.82 to validate the null hypothesis. Since p is greater than 0.5, we can conclude the null hypothesis is valid. There is no statistically significant difference between observed and expected. We do conclude the coins were flipped randomly. The last question, in dihybrid pea-plant cross, in a dihybrid pea-plant cross, pea-plant offspring were counted. The following was obtained. You got 340 tall axials, 115 tall terminals, 105 short axials, and 37 short terminal. Now this was a big A little a by, or big B little b by big A big B little b cross. Okay, it's asking you to convert those to a whole number ratio. So all you have to do for that is divide all numbers by the smallest number, so that you can say that a ratio here. So 340 divided by 37 rounded to the nearest whole number is 9. 115 divided by 37 rounded to the nearest whole number is 3. 105 divided by 37 rounded to the nearest whole number is 3. And 37 divided by 37 is 1. The ratio comes out to 9.331. What this means is that these were 9 times bigger than this. The tall terminals were 3 times bigger than the short terminals. The short axials were 3 times bigger than the short terminals. And the whole ratio was based on the short terminal, and that's why that's a 1. All right, so if you have more questions on these, please ask in class. And please go over these questions before you walk in to take the test.